Hey, you know that stuff hasn't actually been proven yet?
Wait, really?
Yeah, there are only 2 anti-aging interventions that have been proven to be effective for research, one of which is caloric restriction.
What's that?
Good question. What is caloric restriction?
Caloric restriction is the practice of limiting one's caloric intake while still maintaining a nutrient-dense diet. Caloric restriction, or CR, as it's often referred to in the field of gerontology, was first recognized as having a potential impact on longevity in 1935, when Clive McKay found that reducing the food intake of rats caused them to live significantly longer. Since then, CR has been found to consistently extend the lifespan of a wide variety of organisms, including diverse strains of mice and budding yeast.
First, let's talk about CR and yeast. One study found that CR was associated with increased lifespan in yeast, as well as increased levels of hydrogen peroxide in the yeast cells. Hydrogen peroxide is one example of a group of molecules known as reactive oxygen species, or ROS, which are highly reactive molecules containing oxygen atoms. They're sometimes produced in our cells, but they can be very dangerous due to their high reactivity, especially since they'll often react with the first molecule they come in contact with. This can be especially dangerous if they react with DNA, since this could result in DNA mutations, which could lead to cancer.
That sounds bad. Remind me again why caloric restriction is supposed to be good for you?
Well, it's not quite that simple. It's widely thought that oxidative damage is one of the main factors contributing to cellular aging, but some evidence suggests that a small amount of oxidative stress can actually help our cells to boost their stress responses, which can slow aging. Sure enough, the researchers found that the increased hydrogen peroxide boosted the activity of 2 enzymes involved in the oxidative stress response in yeast. These enzymes were able to decrease the levels of superoxide anions, a more reactive type of ROS, in the yeast cells. And the researchers found that damage to proteins in the yeast cells decreased over time in calorically restricted yeast compared to control yeast, and this was also associated with the increased lifespan in the CR yeast.
Wow, that's super cool. It's really interesting that reactive oxygen species, which we thought were dangerous, can actually be beneficial.
Exactly. This is a principle known as hormesis, also referred to as the Goldilocks effect, in which a small amount of something may have no effect or a minimal effect. A large amount would be harmful, but a medium amount is just right.
That's awesome. But this is just in yeast. How do we know the same is true for humans?
You bring up a good point. Yeast are very different organisms from humans, so it's not entirely clear whether these results will apply perfectly to human aging, but the authors believe that the mechanism causing extended lifespan from CR in yeast is likely conserved in humans and many other organisms. The idea of hormesis could also be relevant for CR in humans because excessively restricting nutrient intake would lead to starvation, but finding a balance where just enough stress is placed on the cells could potentially have beneficial effects on the human lifespan.
Now what about caloric restriction in mice? Well, in a study done at Boston College, the researchers wanted to know if the changes in the biomarkers of caloric restriction found in blood plasma of mice were similar to the changes found in humans during a very low calorie diet or therapeutic fasting. They first organized the mice into 2 groups. One group that was unrestricted in what they could eat, and a second group that had a 40% restriction, but they then took the second group of mice that had a restricted diet and allowed them to eat unrestricted again for another 3 weeks. After this additional 3 weeks, the plasma was then collected for the second group again.
Wait, why did they allow the second group to return the unrestricted feeding for so long?
Well, there are not many studies that have looked at the longer-term effects of caloric restriction or fasting on these plasma biomarkers after normal feeding is established again. They found that at the end of the first 3 weeks, the glucose levels were 41% lower in the restricted mice than in the unrestricted mice. They also found that the levels of fat like ketone bodies were 367% greater in the restricted mice than in the unrestricted mice. From this, they concluded that the reduction in plasma glucose and cholesterol, combined with the elevation of ketone bodies and fatty acids, are robust biomarker changes for caloric restriction in mice. However, upon unrestricted refeeding to the second group of mice, all of these biomarkers returned to the levels observed prior to caloric restriction, except for the amount of food intake and glucose levels.
What does this mean for the mice?
Well, this could mean that these mice who have undergone a period of caloric restriction but returned to normal food intake have established a new, more efficient homeostatic state, where they are able to maintain the body weight they had prior to caloric restriction, even with a reduced food intake. This suggests that the mice are better at producing energy by burning fats through ketone bodies instead of relying on sugars like glucose.
Yeah, but what do these results in mice have to do with us?
These plasma biomarker changes that were observed in mice are similar to those observed in humans during very low calorie diets or during therapeutic fasting. This shows that moderate caloric restriction in mice mimics a low-calorie diet or therapeutic fasting in humans. Therefore, the numerous health benefits documented in mice following caloric restriction may also be experienced in humans on a very low-calorie diet or during periodic therapeutic fasting.
Finally, let's talk about caloric restriction in humans. In a study at the Pennington Biomedical Center in Baton Rouge, Louisiana, researchers hypothesized that sustained caloric restriction over 2 years can induce metabolic slowing in healthy adults. Now, why are they studying metabolism? Isn't the point of inducing caloric restriction to study longevity?
Yes, but unfortunately, this isn't so simple. First, following a restricted diet is hard, and very few people would want to do this for a long period of time, let alone their whole lives. Second, in laboratory animals, it's already difficult to control the experiment to isolate caloric restriction as the main variable impacting longevity, and doing so in humans would be pretty difficult since there are so many other external factors that could affect longevity. Also, humans live a long time, and sustaining a study over several decades would be incredibly expensive and time-consuming. So, while it's unrealistic to directly test the impact of CR on human longevity, we can test its impact on other biological systems and pathways that are thought to affect aging, which gives us an idea of how sustained CR might influence the lifespan. For example, two key theories of aging include the rate of living theory, which proposes that mammalian metabolic rate is inversely related to longevity, and the oxidative damage theory.
Given that CR has been demonstrated to extend lifespan across a variety of organisms, the researchers were testing the hypothesis that CR would result in reduced energy expenditure and endocrine mediators in humans, reflecting a slowing of metabolic rate and consequent slowing of ROS production that would support the rate of living and oxidative damage theories of aging. The goal was for participants to achieve 25% caloric restriction, but the participants only ended up adhering to a 15% reduction over the full 2 years. Still, when the researchers measured the subjects' metabolic rate and biomarkers of both metabolism and oxidative damage, they found that the participants did experience sustained metabolic slowing and reduced signs of oxidative damage, supporting both of the targeted theories. Let's look at some of the data. They measured both sedentary energy expenditure, or 24-hour EE, and sleeping energy expenditure in 19 control subjects and 34 calorically restricted subjects. As seen in the graph, 24-hour EE was decreased in both groups, but sleeping EE was significantly reduced only in the CR group. Levels of excretion of a biomarker of oxidative stress, urinary F2 isoprostane, were also documented and found to be reduced only in the CR group. Taken together, these data support that caloric restriction can induce metabolic adaptation and reduced oxidative stress in humans. And according to the rate of living and oxidative damage theories, this suggests that caloric restriction may also have the capacity to extend human lifespan. However, this study does have its limitations. The experimental group was small, only 34 people, and majority female, making it difficult to generalize the data to the larger global population. The study also took place over a relatively short period of time, only 2 years, so we have no idea how caloric restriction may impact metabolism over the course of a lifetime.
Now that we've seen the evidence, the next question is why and how caloric restriction helps you live longer. It comes down to how your cells respond when they sense that nutrients are low and the nutrient sensing pathways that shift the body from growth to repair. First, caloric restriction lowers insulin IGF-1 signaling. This reduces the activity of the PI3K AKT pathway, which normally drives cell growth and division. When that signaling drops, the cell redirects its resources away from making new proteins and towards stress resistance and maintenance. The second pathway is mTOR, which stands for the target of rapamycin, which is a central nutrient sensor. When food is sufficient, mTOR promotes growth and blocks autophagy, but under caloric restriction, mTOR activity decreases and autophagy is activated. Autophagy is a cell's internal degradation system. It breaks down damaged proteins and dysfunctional mitochondria. Instead of constantly building, the cell is doing quality control. In summary, caloric restriction works through two conserved nutrient-sensing pathways, insulin IGF-1 and mTOR. When they're downregulated, cells shift toward repair processes like autophagy. At the same time, caloric restriction slows metabolism, lowers oxidative damage, and can lead to a hormetic response that boosts antioxidant enzymes. These mechanisms explain why caloric restriction has consistent effects across yeast, mice, and humans in terms of improving biomarkers of aging and extending lifespan in model organisms.
Wow, what does all of that mean for me? Should I start eating less now?
Well, it's not quite that simple. We've seen some evidence that CR can extend lifespan in humans and other organisms, but that doesn't mean that everyone should just start eating less. According to current research, it seems like a 40% restriction of caloric intake is most effective, though this may be difficult for people to realistically maintain, so 10 to 25% might be more reasonable. It also depends on your total daily energy expenditure, meaning that someone who already expends a lot of energy, aka burns a lot of calories, such as through heavy exercise, should not restrict nutrient intake as much as someone who exercises less. Also, people who are young and still growing should not restrict caloric intake, as sufficient nutrient intake is necessary for healthy growth. CR could also have negative side effects such as irritability, fatigue, brain fog, difficulties with thermoregulation, and even loss of bone density. In general, CR probably isn't a good idea if it makes your daily life really unpleasant, in which case it's probably hurting more than it's helping. It's important to find a balance that promotes lifespan extension while minimizing harmful side effects. There are also some less extreme interventions that could have similar benefits, such as intermittent fasting and time-restricted eating.
Great, thanks for explaining all that to me. No problem. Hey, did you forget about everything we just talked about?
No, of course not.
Then what are you doing?
Look, that science is super cool and all, but I just don't think it's worth giving up all this.
Well, honestly, I can't even argue with that.